Hello, my name is Joshua Haynes, and my project was on inside the biological, neurological, physiological, and genetic makeup of Alzheimer's disease. Here's the outline that I will be using for my presentation. So the first question is, what is Alzheimer's disease? Alois Alzheimer, a German psychiatrist and neuropathologist, was the first person to discover Alzheimer's disease. This disease was named after him. Here's an image of him. Alzheimer's disease is a progressive disease of the brain that is characterized by the impairment of memory. It's also a form of dementia. My objective of this presentation is to increase the awareness of Alzheimer's disease and present current research on the biological, neurological, physiological, and genetic makeup of Alzheimer's disease. Currently, Alzheimer's disease is a popular topic on the media. Here are some images of magazines that have focused on Alzheimer's disease. There are two primary lesions associated with Alzheimer's disease. There is the neurofibrillary tangle and the senile plaque. The loss of synaptic components is a change that clearly has a significant impact on cognitive functions of the brain. According to the NIH, short for National Institute of Health, ApoE4 is called a risk factor gene because it increases a person's risk of developing Alzheimer's disease. Finding Alzheimer's disease symptoms early can help people start treatment and new interventions to increase their lifespan. Here are some pertinent facts about Alzheimer's disease. Every 67 seconds, someone in the U.S. develops Alzheimer's. Also, 1 in 3 seniors dies with Alzheimer's or another form of dementia. More than 5 million Americans are living with Alzheimer's disease currently. Almost 2/3 of Americans with Alzheimer's are women. Women are at the epicenter of the Alzheimer's crisis. There are 3 known stages of Alzheimer's disease. The first stage is known to be the early stage, the second stage is the moderate stage, and the third stage is the severe stage. The early stage is the mildest form of Alzheimer's disease. Some examples of the early stage are memory loss in conversations and repeating questions. The second stage is the moderate stage. And symptoms of the moderate stage are confusion of current events or time, persistent memory loss, or in need of reminders and assistance with daily activities. The third stage is the most severe stage. This stage includes symptoms of loss of recognition of familiar people or family or places, total loss of verbal skills, unable to care for themselves, and of course the patients need total care. This will last for approximately 1 to 3 years. Understanding the significance of these age-related changes begins with knowing the difference between what is normal and what is an early symptom of Alzheimer's. Here's a table describing the signs of normal versus Alzheimer's symptoms. There are 5 A's to diagnose Alzheimer's. The first A is anomia, then apraxia, agnosia, amnesia, and aphasia. Alzheimer's is usually diagnosed clinically from the patient history, collateral history from relatives. They're also used in advancing images such as CT scans, MRI scans, SPECT scans, and PET scans. Some medicines used for treating Alzheimer's disease are Aricept, Celexa, Depakote, Exelon, and Namenda. These are all used to reduce depression and anxiety, treat severe aggregation, prevent or slow the symptoms of Alzheimer's disease. Currently, there is no cure to prevent or stop the progression of Alzheimer's disease, but there is promising research being performed on this field of study. 3 possible ways to stay healthy are staying physically active, adopting a brain-healthy diet, and remaining mentally active. In an Australian study, the purpose of this study was to identify biomarkers that can predict reliability which individuals are likely to develop Alzheimer's disease and over what period of time. The methods used are, the first method was to get individuals to use for tests. So 4,000 individuals responded from the survey, and from these 4,000 individuals, 1,166 individuals were selected. Once they were selected, they were all categorized. There were 768 healthy individuals, 211 Alzheimer's disease patients, and 133 mild cognitive impairment patients. Out of these patients, 287 of these individuals were imaged for MRI scans, PET scans, and they were imaged on these machines to collect their data. They also had low radiation scans and also accessed body composition such as fluid, bone, and adipose tissue. Now, all these individuals were monthly clinically reviewed and were performed with AD, MCL, and healthy patients. So AD was Alzheimer's disease patients, MCL were other patients, and healthy patients were, just normal healthy. MCL stands for mild cognitive impairment. So they had a little sign of cognitive problems, but they weren't labeled as Alzheimer's disease patients yet. And after all of these patients were monthly clinically reviewed, they were statistically analyzed according to data elements, normality, and distributions. So here's a picture of the age frequency distribution of the three different groups. As you can see, the healthy control group was much larger than the participants with mild cognitive impairment. The Alzheimer's disease groups combined contained a substantial number of very elderly healthy participants. Here's another figure of the data used for this study. And the only difference between these groups was observed in the weight measures for Alzheimer's disease patients. They weighed less than both the healthy controls and the mild cognitive impairment. This slide presents self-reported current and past medical histories of the individuals studied. So, in conclusion for this study, participants with Alzheimer's disease had neuropsychological profiles which were consistent with Alzheimer's disease and were more impaired than participants with mild cognitive impairments or healthy controls. Another study that was done was called Forecasting the Global Burden of Alzheimer's Disease. The goal of this study was to forecast, of course, the global burden of the disease and evaluate the potential impact of interventions that delay disease onset or progression. Here's a model that was used to forecast the worldwide prevalence of Alzheimer's disease in this study. The methods used for this study was to first derive a formula that was relating to age or the early stages of Alzheimer's disease. And then after that, they forecasted diseases by looking at the demographic population of the United Nations. Once they did that, they used published incident rates of Alzheimer's disease and the estimate ages that these groups had. And once they did that, they fit it into a linear line of regression equation. After that, they modeled the death rate of Alzheimer's disease and collected and analyzed the results. So this data estimates that 48% of cases worldwide are in Asia, and that the percentage in Asia will grow to 59% by 2050. In this graph, the prevalence rates at ages 65, 75, and 85 years were 90%, 42%, and 14.7%. This data estimates that about 62% of worldwide cases are females, reflecting the lower background mortality rates among women. According to the American Medical Association, prevalence is expected to increase almost to 13.2 million persons in 2015. Patients taking either lovastatin or pravastatin had a prevalence of diagnosed Alzheimer's disease that was 73% lower than those taking other Alzheimer's disease medications. So this is very positive data for the results. Here's another graph figure that showed that patients taking lovastatin and pravastatin helped the 73% that was lower for Alzheimer's disease medication treatments. Alzheimer's disease is the 6th leading cause of death in the United States and is usually diagnosed clinically from patient history and collateral history from relatives. The benefits of finding Alzheimer's risk factors early can help identify people at high risk, especially females, so that they can benefit from immune interventions and treatments immediately. And here's my works cited page. Thank you.